Hello and welcome back to NEET Decoded. Hope you have gone through the explanation of the previous videos in the NEET MCQ series, and in this video a new question will be discussed. So let's start. The question is, match the organism with its use in biotechnology. The options in column A are A. Bacillus thuringiensis, B. Thermus aquaticus, C. Agrobacterium tumefaciens, and D. Salmonella typhimurium. Now in column 2, the options are 1. Cloning vector, 2. Construction of first recombinant DNA molecule, 3. DNA polymerase, and 4. cryoproteins. Now Bacillus thuringiensis is a gram-positive bacteria which produces insecticidal protein, the cryoprotein. This protein is produced in inactive form. Now, the inactive protoxin, upon entering the gut of insect when it ingests it, gets converted to the active toxic form. Now, the alkaline pH of the gut is responsible for this change. You can see here. Here, the alkaline pH converts the protoxin into the active toxic form. Now, the mid-gut epithelial cells of the insect gets damaged, causing its death. What happens that this cryotoxin is taken in by the specific receptors in the mid-gut epithelial cell membrane. And once that happens, the toxin enters the cell membrane, damaging it. Here, pores are formed, and finally, it leads to the death of the insect. So, the Bt toxin is coded by the cry gene. There are many cry genes which encode proteins that control specific insect pests, right? Now, Thermus aquaticus is a thermophilic or heat-loving bacteria. It is found in hot springs. It was first of all isolated from the hot spring of the Yellowstone National Park. Now, you can see here in the picture. Now, from this bacterium, a heat-resistant enzyme, Taq polymerase, is isolated. Now, this enzyme is actually used to amplify DNA. That is, multiple copies of DNA are made from this. This enzyme is thermostable and very active and very useful in amplifying DNA. So, the enzyme DNA polymerase is obtained from Thermus aquaticus. Right? Now, Agrobacterium tumefaciens is used for DNA transfer in plants. Now, the TI or tumor-inducing plasmid is present within the cell of the bacterium. As you can see here, this is the TI plasmid and this is the bacterial genome. The TI plasmid is capable of dividing on its own, independent of the bacterial cell genome. Now, the transfer DNA of the plasmid gets integrated into the host plant cell. As you can see here, it is integrated here. Now, this is used to deliver the desired gene into the host plant. Now, this capability of agrobacterium is used for genetic manipulation. What happens that crown gall is formed as a result of transfer of the gene. So, any useful gene or desired gene is transferred using this vector. So, Agrobacterium tumefaciens is basically a cloning vector. Now, Salmonella typhimurium is a bacterium carrying a small circular DNA or plasmid. Now, antibiotic resistance gene was cut from the plasmid of Escherichia coli and inserted in the plasmid of Salmonella typhimurium to create recombinant DNA or rDNA. You can see the plasmid here. And this is the bacterial chromosome, a bacterial genome of Salmonella typhimurium. Now, as has been discussed, the antibiotic resistant gene was taken from the plasmid of Escherichia coli and recombinant DNA was created. This was initially or first of all done by Cohen and Boyer. So, the answer is option number 2. You can see in this column, Bacillus thuringiensis for cryoproteins, Thermus aquaticus, DNA polymerase, Agrobacterium tumefaciens, cloning vector, and Salmonella typhimurium for construction of first recombinant DNA molecule. Fine. So please like, share, and subscribe to my channel. And thanks for watching.